talk I'm going to do today is try to summarize the thing I've been doing since 2018 and so, and then mainly I'm going to cover the concept of which I call the manifold fitting, and then also I will add at least two big applications that we have been doing since 2020. I think the pandemic made most of people very boring, so we have been, not only doing this methodology development, but also trying to work on these data. And eventually my students and I, also the postdocs, they actually wanted to open a company with me. I don't know about how things are going to turn out, but the two application will be using manifold fitting to pre-process very big data. And I'll try to make sense why do we actually wanted to do this. Okay, I'm a statistician, I actually got my degree, in Pittsburgh around 2011 or so. And then I think 10 years before I graduated, also 10 years after I graduated, I think there are a huge number of people in my field, in statistics has been doing dimension reductions. And around 2014, I was trying to think about this and how effectively can we actually do dimension reductions and what is the room that we can improve upon. And this is what I call, manifold fitting. And we'll see how, whether it is even useful to actually do things like that. All right, so this is try to connect with the statistics I've been doing and also with the geometries. I call this as a new landscape because I think that after the pre-processing using manifold fitting, then things will get much easier to analyze. If you also show the difference between either you fit or not fit about this high dimensional data. All right, so from now on, when I say high dimensional data, I mean just any data format like n by p matrix. All right, and then we're trying to do things on the n by p matrix. All right, so this is just a very high level, things that around a few years back, and I saw this called Sora, and they come up with this open AI things, and they try to. What they really do is trying to generating things. You can generating pictures, generating images, sound, and all these things. What they do, from my understanding, is basically you've actually taking all these data. For example, you collecting all these images, collecting all these sound, pre-processing them, and the philosophy is that you try to take the high dimensional input and then map them, or maybe I should use a word called embed them or encoding them onto a very lower dimensional structures. This is a space that when you try to do these embeddings. And then whatever people have been doing, basically they're doing business in this very lower dimensional space. Different people use different ways. Sometimes people call it a lower dimensional space. Sometimes people call it a lower dimensional manifold. Sometimes people say it's just it is a space with kind of a sparsity on it. And then you can do clustering. You can do all these downstream analysis by getting a loss function. Somehow what you do is just try to minimize the loss functions. And then you wanted to decode them and be basically going back to the original space. This is how things has been doing, at least from my point of view. All right, so you need to decode it back to the original space to see how this thing looks like. From my point of view, every task that people have been doing on this lower dimensional space or latent space and so on, right? These are all attached to the downstream analysis task. Whatever function that you are trying to optimize, let's say clusterings, classifications, or whatever, they are attached to the downstream analysis. Because of that thing, you wanted to optimize. If you have a different task, you're getting a different latent space to embed your data onto that space. So is it truly intrinsic, that space that people do business upon? Or can we actually get a lower dimensional space or lower dimensional manifold to actually map your data onto that space? Hopefully, that space has nothing to do with this downstream analysis. So once I saw this, for example, the Sora generates this kind of pictures. Of course, for this applications, you generate things. In this case, the red wines, coming out of the glasses, and we basically think that it might be still room for us to improve when we try to, summarizing things onto that lower dimensional space. So from now on, what we're trying to do is we're trying to find a lower dimensional structure basically I actually call this as a lower dimensional manifold behind a high dimensional data. The data living in high dimensional space, and no matter how you see it, it's a picture. If you line them on, it's going to be a high dimensional vector. So the point living in a high dimensional space, what we're trying to do is push that data onto the lower dimensional manifold behind it. We never know the manifold, okay? But the manifold living in the high dimensional space. To differentiate, from the thing that we do with the dimension reduction is that we're not really selecting a few variables out of, let's say, 10,000 variables. We will actually see every high dimensional input as a point. So basically we have a huge number of the point cloud that we try to learn a lower dimensional manifold so that we can push. When I use a push, I mean we will actually project all these points in its original ambient space to the underlying manifold as close as possible. If we can do that, then we can do many things on that manifold. And because of manifold living in a high dimensional space, so you don't need a decoder to come back to your original space to see how things goes. That's our somehow, the way of thinking. Okay. All right. So before I talk about the manifold fitting, because we're still we have still some audience who are statisticians. I just wanted to review some concepts. The PCA has been used for from my point of view, maybe at least 60 years. Everybody, if you really double check all these pipelines that that people has been building, and they publish papers, or they're actually doing all these very fancy, I would say, the analysis for large amount of data. PCA basically is the backbone. What the PCA do, from my point of view, is that you've got a data cloud, right? In this case, it's just a 3D. Think about we have a bunch of data in R3. Then what the PCA does is that you actually start from, let's say, the mean of it, sample mean. You compute the average of the samples. This is what this the blue point is actually about. Then PCA somehow do the eigen decompositions, or you can do SVD and so on. You will get the first largest 3 eigenvectors. And then this is basically the A1 or A2 and also the A3 here. If you look at PCA in a backward way, like Steve Murray has been doing, PCA essentially will generate a nested subspaces. And for this particular case, it's the cube it's essentially a subspace with dimension 3. And that cube contains this green things, which is 2-dimensional. It's just a piece of paper. Then that piece of paper actually, is basically a span of the first 2 eigenvectors and so on. And then this cube contains the green things. And then the green things contain A1s. And A1 contain this blue dots. So basically, if you look at PCA in the backward way, PCA basically generates a sequence of nested subspaces. And in this case, the PCA, all these subspaces are linear, that's all. And people call it as the flat space. Okay, that's how I actually see how the PCA behaves. And around 2013, when I was a postdoc in Switzerland, I actually saw this paper, which was also Sankyu and also Steve Merrill has been actually work on this. So basically what they do is that from my point of view, of course, in order to actually generate another type of the flat space, they somehow deform the data. Suppose you have high dimension data, you deform the data so that the points will be eventually lie on a hypersphere. So you have a long vector, you normalize it basically. And you've got the points in hyperspheres. All right. Then you keep slicing the hyperspheres using Euclidean space. Every time you slice the space, you've got map all these points on the hypersphere to the circles. And then you can map the circle to another sphere with dimension less than 1. Sorry, with dimension 1 less, and you keep doing this. So this is called principal submanifold. Sorry, this is called the principal nested spheres. So it generates a sequence of nested spaces. But in this case, all these spaces are still circled spheres, which is nonlinear, of course. Around 2014, I was thinking maybe I should do something, to extend this. So my original idea was maybe I should, maybe I should to generate a sequence of nested submanifolds where each of them actually was contained by the other ones. For example, I can generate, let's say, M0 up to n d minus 1, d minus 2, and so on. Then the d minus 1 is a submanifold contains the m d minus 2 and so on. So you can actually view this d minus 1, d minus 2 as just an analog of the previous, the flex spaces. And for this case, for the principal nested spheres, these are just spheres. All right, for the PCA things, the flex space is just linear subspace, just lines to me, hyperplanes. For this one, I will call this as a submanifold. Then in order to get every single of these m with dimension something belong to, from 1 to d minus 1, that every single of these are actually manifold. In other words, if I really wanted to generate the sequence of the flex space like this, I need to have a technique to fit the data. Be careful here. I'm not actually normalize my data onto a subspace onto a spheres. I'm just fitting the data to get this manifold, and then fitting the data with to actually get this manifold. I call the process of fit the data with this dimension as the manifold fittings. In other words, we're trying to get this submanifold so that, yeah.
I'm just wondering, are you thinking about doing this in a forwards way, a backwards way?
It doesn't matter.
And fit the whole flag?
It doesn't matter.
Doesn't matter.
Yeah, I can do it in the black ways also. So if I can get this, if I can get, let's say, K or D minus 1 submanifolds as estimators, and I can guarantee that every single estimator is contained by the other ones with dimension 1 higher.
When you say it doesn't matter, do you mean you can do either one, or it gives you the same answer both ways?
I can give it, well, for me, I can fit a sequence of these manifolds.
Either way?
Either way.
But it doesn't necessarily give you the same answer both ways. So if you make increasing ones, it might give you a different sequence than if you take decreasing ones.
For this techniques, the only thing I need to know before I actually do the fitting is the dimension of the submanifold I actually wanted to fit. So I wanted to introduce this something called manifold fitting to fit each of these guys. Okay. Well, back to 2014, I didn't have this technique called this manifold fitting, so I don't know how to do it. So everything I've been doing is trying to extend that's PCA to a known manifold, where we basically know the geometries, something called principal flow and so on. But here, if we've got data sets, okay, you can view every single data set like data cloud. So I can, so basically I'm trying to fit a manifold with different dimensions to the data. And specifically to guarantee that all these manifolds are nested with each other. And yeah.
Somehow optimize that these manifolds will be as simple as possible?
It depends on curvatures. There's no, actually there's no unique solutions for the fittings.
Okay, so choose.
We're not optimizing anything. We're trying to find a meaningful manifold to project the data to make the downstream analysis independent of all these things. We're not optimizing over a perfect manifold. Yeah. All right, so as I said, I will actually try to talk about this manifold fitting and also introduce 2 recent big applications. This has something to do with my postdoc and also PhD students, and also a joint work with my senior co-author and also mentor, STLs, who actually founded this the center called the CMSA. All right. So the toy problems. You've got all 3 data. Okay, these are the points in all 3. They've got coordinates and so on. So you wanted to do clusterings, for example. So a very common way that people do it is just, well, this is all 3. They're just going to flatten the whole Swiss road. Meaning that you will actually flatten the data sets to map them onto the points on the 2-dimensional planes by preserving some distances. Well, that's okay. And there's a lot of ways to do it. People call this process as dimension reduction. Once you project all these points onto the 2-dimensional planes, you can apply all these Euclidean methods to do clustering, to do all these downstream analysis. Well, even if you can preserve the distances between the two different spaces for the points, you actually destroy the geometries. So the thing I wanted to do, which I call manifold fitting, is given all these points, I want to fit the surface in R3 space. And the surface I'm actually going to fit is just the Swiss road. If I can do that, I can project every single point on that Swiss road, fit the Swiss road. And then I can do all these analysis in R3. Questions so far? So in other words, I'm not going to cut down the dimensions. I'm going to learn the Swiss road surface and then map them onto that surface. Yeah.
Then how do you overcome the curse of dimensionality?
Why do I need to overcome?
Because it seems like your ambient dimension is still too large to do like good algorithms.
For the R3 case, the ambient space dimension is extremely low. It's 3. Later on, I'm going to talk about this, the big application, the ambient space is 15,000 to 45,000. So what I do is I don't cut down the dimension, as I said. I will try to push the points in its original space. But the manifold itself has dimension very low.
Starting from here, flag, start with dimensional manifold fitting.
No. The flag is basically says that if I can get every single manifold, but here I'm only saying fitting one of these guys. That process, I call them as the manifold fitting.
Where do you
I was just gonna ask if you select the dimension first, or you let the data tell you which dimension to
That's a good
use?
question. Before 2019, I need to know the dimensions, the intrinsic dimension of the manifold. But now I don't have to know. I don't have to know. So again, this is also back to 2016-14. I was trying to extend the PCA where I actually know the manifolds of the geometry. For example, we talk about the earthquake data, X, Y, Z's. You can treat them as a vector in R3, but also when people try to actually study this kind of things, they want to fit a line or fit a curve, let's say gamma, traveling around this data set, but also the gamma has to be on this S2. So in order to fit a curve, for example, I'm starting from somewhere in the center and I'm actually going to get a gamma. The gamma has to stay on the manifold, not going to upside and down inside this whole spheres. That's just a way to extending the principal component to something called a principal flow, where we do need to know how to compute things. We do need to know what the manifold is. In this case, the manifold is S2. If you model the Earth as a sphere. The toy problem, if we've got a multivariate data, right, N by P matrix or P by P matrix, and this is just saying if the multivariate data N by P, that if we have some constraints on the data sets, then basically we're actually dealing with a manifold situation. And this is just a very naive example. Again, we're still talking about toy problems. Covariance matrix, 3 by 3 matrix. It's 9-6 because we've got 9 elements. Because of the symmetry, if I let the P3 be a space of all the PSD, defined by this 3 by 3 matrix, then because of the symmetries, the P3 is essentially is a subset of R6. But R6 is still too big for us. Then actually P3 is convex, but actually not linear if we have got 2 covariance matrices. Then you can view them as 2 points on some kind of manifold. The combination of these guys might not actually in P3, meaning that the P3 is not actually closed. There's 2 ways you can choose. One is you just treat them as a R6 vectors, 2 covariance matrix. You apply all these Euclidean space methods doing optimizations. The second choice is that you can view all these 3 by 3 matrix as a point on a cone, at a cone living in R4. All you have to do is develop a ways to analyze all these points each point representing a 3x3 matrix on the columns. Which means that you have to actually extend all these methods that you actually use in Euclidean space to the column. Again, this is still the geometry we actually know. Okay? This is a bit more ambitious. Suppose we've got high dimension input of the images. Every single point here is an image, is a very high dimensional vectors. You can think of it as, can be a 3,000 dimensional vectors. So if we want to do clusterings or classifications, what people have been doing is that they will apply either the activation functions, layers, try to transform these points from its original space to some other spaces, determined by the function you use. In deep learning, you can actually try to transform all these things to a very complicated space, and you do the analysis in that space, and you come back here to get the boundaries. What I would do is basically using all these points in high dimensions, images, I will try to learn the manifold. And then if this manifold can be learned, then I will actually do a boundaries directly on the learned manifold and see the differences. There are two ways, as I said. Okay. So I just wanted to mention a few things. I've actually checked on a fairly large number of the literatures, and when I was trying to learn this, actually, when people talk about the manifold learning, and I think Charles Pfefferman tomorrow is going to give some overview about this. But I want to make a distinction about my understanding of the manifold learning with the thing that I actually learned. So people assume that if you have a high dimensional data in RD, if there's a manifold behind it, the data is very noisy in RD. If you believe, or if the assumption of the manifold, the lower dimensional manifold is that they're actually behind this high dimension data, living in the same ambient space. What all these manifold learning approach has been doing, from my point of view, okay, starting from 1990 something to 2010, are essentially dimension reduction methods. They reduce the dimensions from capital D to small d. He mentioned this so-called personal dimensions. This is very big. And if you assume there's an underlying manifold with a small d, what the people do is basically estimate embeddings. They output this RD space. That's all. I don't think all these methods actually estimate the manifold. That's why I call this as a dimension reduction methods as manifold learning. I don't know why, but I've been looking at all these papers. They do not necessarily estimate the manifold. What I wanted to do is that if there is an underlying manifold, and with a very, very small d, I want to actually estimate the estimated one, and somehow guarantee that the dimension of that thing is roughly d. I want to give a manifold estimate and control the underlying manifold, which we never know with this guy, in terms of some distances. I will not suffer this approach. If you can fit this manifold, you will not suffer the so-called curse of dimensions. In other words, if you view a data cloud in very high dimensions, all I do is project that data point in its original space to the underlying manifold as close as possible, and guarantee the distance between these two guys with high probability. Okay, so for example, ISO map all these local linear embeddings, Haitian version of it. Even my friend invented this LTSA, local tangent space alignment, and so on. They do not actually estimate manifold. They just output this R small d things, give you a much shorter vectors. Of course, you can do optimizations by telling me that upon any other downstream analysis, you can do the best. But if you change the downstream analysis, the latent space is different. So I actually, around 2016, I actually looked it up, and I saw this picture. This paper was very interesting. The 2012 There's two papers. Actually talk about this fitting problems, but they don't actually give a very concrete algorithms. They propose some ideas. And then I noticed that C.F. Pfefferman and also his co-authors did something on this 2016, posed a high-level thought about this manifold estimation problems. And I believe tomorrow he's going to talk about this. And 2018, they have a much more concrete, method telling us, is it even possible to actually do that? And then myself and also my postdocs, we did something on 2019 and trying to improve upon his methods on, based on this 2018 papers. And 2022, we had a paper trying to improve some bonds, improve the bonds between the distance between these two guys. And then after that, there's a huge number of the applications that my student has been very interested in. And we applied this thing to a single cell. And we applied this thing to the so-called metabolism data set, which I bought. We spent 9,000 pounds to actually bought the usage of the Euclid Biobank. And I tried to release this results by the end of this talk. This is the first single cell application we tried. And back to 2022, I was visiting CMSA for a year, and someone from the HMS, Harvard Medical School, told me, if you really wanted to sell this method, you want to do very applied jobs so that people can use it. So this is the collection of 25 data sets from the single cell data sets. All right. So the number of the genes means the dimension of the data. The number of the genes goes from 15K to 40K. This is pretty big. In other words, we've got 20 rows here, every row representing a data set. The first one has 704 samples. The number of genes is between 30,000, so on. Roughly 38,000 is actually very big. According to the technicians, they determine somehow by some method, okay, these are the number of the cells, the number of the type of the cells. So later on, we'll actually show if I fit my manifold fitting onto these matrix, A by P, A by B matrix, without using these informations, and we'll show how my method will actually produce something that we can actually do the clusterings without knowing all these label informations. In other words, for the first row, we've got 704 samples. I will push all these samples in a high dimensional space, 30,000 dimensional space, in its original space to an underlying manifold, and return you the 704 samples. The thing I'm going to return to you is basically the same matrix, is a matrix with the same dimensions, 704 multiply 30,000. And we're going to do this for every single data set. I'm going to show the results by the end of my talk. And the second application is that the so-called the metabolism data set. The UK Biobank basically follows 500,000 people. So this data set basically examined almost 200,000 people, 200,000 patients. And they do the MRI on the blood samples, not on the head, on the body organs, on the blood samples. If you do MRI on the blood samples, they've got 251 variables. These variables basically are indicating whatever metabolism activities. So you can think of this is a huge matrix, which is almost 250,000 multiplied 251. So we're gonna fit this manifold onto that matrix and try to visualize something. And we'll also report the results by the end of my talk. Questions so far? All right. So mathematically, I think that for me, that I actually translate this the geometric whitney problem to statistical problems. Basically, we'll assume there's a manifold behind this high dimensional data, and then the capital D stands for the dimension of the ambient space, and you can interpret D as the number of genes. or the number of the variable and so on. And there's metric on basically on the M, the supported M is actually M here. So X is noiseless data. We do not observe the X here. If you have some noise on it, then the X plus this C here basically will lift the X to the Y here, the Y living in the ambient space. All we have to do is learn the Y, trying to learn this, give a estimate for called the M hat. If you want to write some papers, you can do some asymptotics so on. All right. Illustrations. This is the dashed line is the manifold, and these are my points. The manifold learning approach will map all these points to a subspace spent by something, and you can determine the subspace upon together with this, your downstream analysis. Once you are here, getting all these red dots, you can't go back unless you have a decoder. For example, if you're PCA, you pick the first k eigenvectors. These k eigenvectors give you space, and you project all these black points, getting all these red points. You can't go back. So what I want to try to do is basically, you can interpret this as actually denoising problems. We push all these points. So arriving at these red dots. The red dots can be very close to the manifold, if we can guarantee that. And then all these red dots living in the same space as these black ones. If I can guarantee all these red dots, which are very close to the underlying manifold, somehow I can get a smooth manifold, and you can do interpolation and so on. Now, this is a very naive version of the model that you can use. That was back to 2019. We observe the data yi's, and xi is basically a point on M. The M is embedded in RD here. By 2019, we assume is all the xi's are the IED samples, uniform distributions on M's. Later on, many people give me some request. Maybe I should not work on IED, but that's a different story. This is a theory problems. M here is a small dimensional, d-dimensional manifold. The previous speaker talked about the reach, and here we assume the flatness of the manifold is with the low bound tau here, and this Ci is a noise distributions. So you've got different noise here, but we are going to target the manifold fitting problems for unbounded noise. So basically, your Yi essentially is just a convolution of the metric for the X and also the noise distributions. Given the Yi is trying to push the Yi's so that we can get all these Y hat basically, so that they can be getting very close to the manifold. So the reason I mentioned this Christian University and also Larry Watson papers 2012 is that they've worked on this. The 2012 paper assumed that the data Xi essentially is, the noise essentially is bounded. And then the noise is orthogonal to the tangent space of Xi on the manifold. So this to me is like you've got a very bounded the observations. And of course, you can somehow basically view this, the setup as a tube, okay? And you can, of course, do this manifold fitting problems. They do get some kind of nice bound, which in this case, for example, a polynomial bound. When they try to attack the problem using a unbounded noise, Gaussian noise, they somehow got this, which is not too surprising because you've got 1 over log n, so you need a huge number of points to actually do this manifold fitting problems. That's why they actually claim that back then, I think, they wrote a blog saying that they're actually not going to worry about this manifold estimation problems. They reduced to some other things called a ridge estimate, just like another estimators to speed up the convergence, to introduce some bias of it. But the ridge estimate is not something I wanted to do. So we're still going to work on the manifold fitting problems where we're looking at the sigma, which is noise level of your data, gets to very, very small. So before I talk about my manifold fitting, I just wanted to mention this 2018 paper that CF has been doing. From my understanding, so this is purely from my understanding, we actually, plot all these things by scratch, by reading his paper. So this is the manifold, and you have, let's say Y here. Y is something you wanted to project to the manifold as close as possible. And what you do usually is that you look around, then basically the Yi, Yj is something you have as a sample points. So all you have to do is find a way to project the things towards the Y star. Y star is something you never know, but the Y star is on the manifold. The Y star is your target, basically. Right? So what you do is actually you look around, standing at the Y here, and at the radius, at the level of the square root of the sigma. It's just trying to make sure that your PCA eventually have some eigengap. The sigma here is the noise levels. So basically, if you've got the sample size and then larger than this guy, and then D somehow is the intrinsic dimension of the manifold. So if you follow this, you can define a level set as a estimator for the manifold. So this is a sample point Yi, then you can somehow estimate the projections at this Yi using the information around this Yi here. So the black arrow is telling you how to shooting the Y using the information that you estimate from the Yi's. And this red one is something you use based on the information you estimate from the Yj's. So eventually you are actually shooting the Y towards the Y star. Then you can add up all these different colored the errors by doing the weighted average. No matter how high the dimension is, that vector is always going to be one-dimensional. So you need one more projections, because this black arrow is not going to really shooting towards exactly Y star, because you never know the Y star. So you need one step projections basically to correct this shootings. So the actual the method that in that paper might be different, but this is something of my understanding is if my understanding is actually correct. So you've got all these Y's in the ambient space. Then after the second projections, if the black arrow, the shooting, the norm of that vector becomes 0, you're arriving at this manifold. So this is a M0 estimate. So you basically control, you can show with high probability that these two guys are bounded. Okay, and R is square root of sigma, so essentially this is O sigma bound. That's my understanding of the 2018 papers. And this is just a very quick illustration of this simple examples. You've got these dots, data sets, right? You've got a circle. I mean, this is not naive problems. All you have to do is, using what they call is epsilon net. You've got all these, circles, Euclidean balls to try to truncate stuff. And then within each of the balls, then you use the data. You can do PCA, you can do whatever you wanted to do. You can get some spaces, which is lower dimension. Then after that, you can forget about all these data sets. These are the estimate directions. So you wanted to project the green dots shooting to the manifold. All right, and then for example, in this case, if you look around at the radius square root of sigma, then you've got 1, 2, 3 different subspaces you estimated previously. You don't have to worry about all these things. They're too far away from the green dots. And then you can do all these weighted average things, and hopefully that you can get the projections, which is just you adding up all these vectors together. And this basically show you. We actually tested these ideas, which is actually okay. Not too bad. Every single point on this picture, 2x2 picture. If I apply that thought, we examine the norm of that vector, the final black arrow vectors. The color means that the light color means the norm is very, very, very small. The blue ones means the norm of the algorithm, that your final projection is big. So of course, for all these points near the circles, the norm is small. So I thought about it in 2018 or 19, and I thought maybe actually, you don't really need to do two-step projections. And the reason is that even if you work with, which I call the unbounded noise, for example, Gaussian noise, you can actually take, let's say, the weighted mean of the data points within the circle, even if you work with unbounded noise. So the Y tutor means the weighted mean of all the points within the circle here. So all you have to do is just do one step projections. And then the reason is that this is the bias the algorithm was introduced. If I can find this Y tutor over here by taking the simple average, I can actually try to estimate the space here as a dashed line here. And that space is always going to be parallel to the actual Y star tangent space. If I wanted to get this X back to this manifold, I can actually shoot it orthogonally towards this dashed space. And this is the bias, which is apparently much smaller than this guy. So this method somehow will speed up or actually bring in some values by constructing the manifold estimators. And we can from here I have to say that we can still even practically we can improve it, but theoretically we'll still get this R squared, which is in this case is actually the sigma. Okay, and there's some details I just wanted to forget about it. In 2022, we somehow still try to improve based upon the sigma squared distances. If the sigma is very, very small, you want to fit the manifold, hopefully to get sigma squared distances from your estimator of the manifold with the underlying manifold. If the sigma is very, very small, so sigma squared is a high order. The previous methods already guarantee that if I'm here, I can shoot this point towards the manifold, arriving at this blue dots. I can guarantee the blue dots is basically O sigma distance from the manifold. That I actually know. I have no idea how. I mean, I don't really care about the manifold, but the blue dots will actually having a distance between this manifold here is O sigmas. If I try to use all these points, let's say under the cylinder constructions, meaning that if I connect these two dots, then construct a cylinders. If I shrink my cylinders, the way that I can project this blue dots further towards the manifold can get this green dots. The green dots will have a high order distance from the manifold. Yeah.
Could you explain again how you constructed the blue dot? It's not on the manifold, is it?
It's not on the manifold. It has a distance which is O sigma distance from the underlying manifold. I never knew the manifold, but I can produce a level set, and then as the estimator for the manifold. I don't know the manifold at all, but the blue dots will be with high probabilities O sigma distance from the underlying manifold, which is already good enough. They're not unique, yeah. But the algorithm will produce something. Of course, the algorithm depends on some other parameters, of course.
Very high D, doesn't that become computationally unfeasible?
For very high capital D or small d,
Capital.
it doesn't matter because that shooting only depends on this dimension of the vectors. The vector is always going to be one dimension. That's why I'm saying I'm doing this inside this high dimensional space. Okay. Here is a very good question. Back to 2019, even to get this blue dots that actually involve the tangent space calculations, that things, if you have, let's say, D around 5,000 doing this decomposition, is already very time-consuming. That's why we somehow using the sort of cylinder calculation that tell us you can shooting the thing directly using the normal space. Oh, sorry, using this the orthogonal space without actually doing the tangent space stuff.
Yeah.
And the reason here is that if the Y star is the target and the Y is something we wanted to project, this Fy is the first step, is actually the first step of the fittings from 2019 papers. All I have to do is control the angles between these two directions. And we can control this angle at the sigma time this very, very small term here. So we don't worry about the tangent space alignment, sorry, tangent space estimation anymore. Because if we keep doing this, the PCA stuff, we're stuck at the 5,000 dimensionality problems. Yeah. So, okay, so I will somehow summarize the thing without putting actually too much attention on the theory because I'm actually running out of time. So eventually we can prove that after the shootings, the two steps, we can guarantee the Y star and also this after the final result is at the rate of the sigma square times a tiny term. Everything else we're gonna do for this real data is based on this two-step method rather than 2019 problems. Otherwise, I would say we can't deal with a matrix which is 10,000 by 10,000 and doing the eigen decomposition. All right, so 2023, I organized the first Harvard conference on geometry and statistics. And there's a very distinguished speaker tell me, maybe you should combine your fitting with the machine learning. So when I came back to Singapore, I thought about maybe I should. The thing is that the machine learning, I actually didn't know too much about machine learning, but I thought about it. He said, maybe you should do something, learn the structure between the two different spaces. So cyclogram is just one of the GANs. The cyclogram learn how to map the Z to the Y's. You can see the Y as some kind of data images, and Z is some kind of a lower dimensional Gaussian noise. The cyclogram is trying to train the two generators, GZ, GYs, and also the two discriminants. Unless you basically by the ending of this whole balance. For anything, for example, in this lower dimensional noise, you can actually get this Y tutor or Y hat and so on. The Y is the real image data. So you can basically generate the fake data and together with the real data. My face, your face, whatever face is. My fake face, your fake face, all these faces. But these are in high dimensions. The cyclogram will pass this to the dy, which is a discriminantors. So I thought about it. If I can fit the manifold, or if I can push all these y's, y tutor, y's, fake image, real image in very high dimensional space. If I can do the fittings, I can actually pull these things onto their underlying manifold before I'm passing it to the dy's. So basically, we just add a simple manifold fitting procedures right there. We don't have to, actually estimate. We didn't put this Fy onto this step because the X is a dimension which is low. Oh, sorry, the Z here is actually in lower dimension space. So we actually train this whole GAN things. And we observe that by introducing this FM here process, we'll actually speed up and also have some very stable results. I guess the reason is that we somehow denoise something in that high dimensional space. All right, so let me come back. So after that, 2014, we've got this single cell analysis. And then recently, my team has been putting up this manifold fitting onto the single cell pipelines, which I call the cell scope. It's going to appear very soon online, but also we are trying to improve it. And then we apply this manifold fitting on this metabolism on biomarker data set. I will try to explain these two results very shortly. And let me just skip up all these details. Oh, maybe I should mention a few things on the cyclogram with the manifold fittings. So these are actually toy problems. All these are the noisy images. You've got L here and rotate L and you've got all these images. Not only the fitting can produce a denoised version of these images, but also if you pick these two images, we can actually extrapolate all these images between these two guys. Be careful. These images, 1, 2, 3, 4, 5, 6, 12 images were not in its original data sets. So which means that we can actually generate things I mean, this is somehow my understanding of this manifold fitting with the cyclogram. Okay, well, the bottom line is that I'm not a expert in machine learning, but it's something I can talk about it. Now, this is the single cell business. This is the first data, 709 or 704, whatever, multiplied 30,000. Okay, it's high dimension. It's high dimensions. And the technician already determined there are actually 3 classes. I don't care, meaning that I'm not going to use this information. It's high dimensions, so some other speakers talk about the t-SNEs and so on. So we're gonna try to visualize the data sets. We've got 704 points, and we visualize them. Basically, we find a way to visualize them so we can see it. This is the results when you apply the t-SNE, which is essentially just visualization methods. Well, you know all these information, so you can try to look at the results. Well, did 3 different colors, so you can see that all these images, also all these points are mixed together. Well, here's just a visualization of the original data set. That's all. Well, people say, oh, why don't you apply some fancy methods like deep learning, autoencoders, try to differentiate these 2 guys or 3 guys called clusterings. Well, you can always do that. Spend some time, some hours, some coding methods. Fine. Hopefully you can increase the accuracy from 57% to 99% or 100%. I mean, that's fine. Well, we didn't do that. We fit this data, return you the same dimension. Let's say return you a matrix 704 multiply 30,000. That's all. It's a fit matrix. And I use t-SNE. Or you can use UMAP or PC, whatever you want to use, to visualize the fit data set. This is what I got. So that was 2022 December. I was very surprised. I thought this is just a coincidence. So we actually double check the codes and so on. So again, we're not doing clusterings. It's up to other people to do the clusterings. We're just showing up the visualizations after the fittings. If you are familiar with clusterings, k-means, whatever, if you can program, you can still apply the clustering methods to class the stuff that I actually got. This is just showing the results, that's all, for the visualization purpose. And you can see that all these points has been isolated. As I said, I thought there was a mistake or coincidence. So we try every single one of these 25 data sets. The previous I showed the 25 data sets that we download from the Nature Science Cells. They were published in the recent 15 years and so on. So we try every single data sets. Some referees insist us to actually compare with the deep learning autoencoder. They say, okay, this paper has been cited 4,000 times. So if you want to publish the result in the PNS, you've got to compare with this guy. Well, we did. So this is the results using this method. If I fit the data without cutting down the genes, and then apply very simple clustering methods, k-means or hierarchy clustering and so on, we've got this. And overall, you can see almost like 25% accuracy, 25% increase in terms of the ARR index. That was the result back to 2 years ago. Now we have a slightly better results.
What do the red and black mean in this table?
The red is that we apply the fitting on the matrix, on the data, that apply very simple clustering methods. The black means that we use this fancy autoencode, the deep learning methods that people use in the single cell business.
See like 4 red entries on the left. I see
Oh,
2 black
because
entries on the
these guys not actually, because maybe these 2, 1, 2, 3, 4 data sets.
Are the red just the?
So this guy, that guy, that guy, that guy means that, okay, for these 4 methods, also for these 4 data sets, we didn't improve too much, but the rest of them overall, we improved a lot. That's all. Yeah, because you can't win every single time. That's all. you've got lose some at some point.
Oh, I see. The red ones are just the ones where you did better, right?
Yeah.
Okay.
In general, we've got 25% increases in terms of the ARR index. That's all. But that was 2022 December results. Okay, so this is just a pipeline. Now we've got a much more formal things that we can do single cells, and the bottom line is that if you fit or not fit, there is a difference. And things that we fit, then we still give you a matrix with the same dimensions. You can apply your own methods and so on. You can still keep doing the thing that you have been doing. And the last one I wanted to talk about is actually the metabolism data set. We've got 251 variables measuring all these metabolism, whatever, levels or things from the blood samples. And we've got 200,000 people. That's a huge populations. Thanks to the the UK Biobank, we can actually use that data set to study something. Okay, so 251 biomarkers. So we split them into 7 different classes. Oh, sorry, 7 different subsets of the variables. So the rows here is a sample, 200,000. Each of the matrix, for example, this matrix is 200,000 multiplied 36, for example, and this is 200,000 multiplied 25 and so on. Basically, if you add up all these variables, you've got 251. So we fit a manifold for every single matrix right there. And we got M1, M2, M3, M5, M7, whatever. So we got 7 results. Okay? 7 manifolds, 7 outcomes. We've got to visualize them. It's, as I said, the group contained of 200,000 people. So we picked we actually picked the 50,000 people and visualize them. This is the original data. But as I said, the data is in high dimensions. We actually use t-SNE to actually put them on the 2-dimensional space so we can see it. There are actually 50,000 people, and we visualize them. This is the original data. So I'm actually showing you the M1. We've got results on M2, M3, M4, M5, 7. I'm going to show you the M1. And this is C1. Now, as I said, you can use deep learnings, whatever you want to use. When you do the deep learnings and try to visualize your results in 2-dimensional space, in this case 2, this is the visualization. We fit the data, get a C1, get the M1. We visualize it. We got this. That was the last year results. People say, so what? You somehow you can get 2 clusters. This thing, you don't have these patterns, but so what? But again, it's a UK Biobank, and every person's profiles. We bought it. So not only the blood samples, all these other variables. There's a huge number of variables for every single person. There are 50,000 people, meaning there's 50,000 points here. The 2 clusters, separate by, at least from the visualizations, and we did this by using this, the metabolism data. But we actually cross-check every person's medical profiles. And we cross-check every person's the profiles. We try to compute, let's say, the risk of the person getting the first 50 most relevant diseases from the medical profile. And we found that for all these people getting the diabetes, high blood pressure, whatever, 50 different diseases, the risk is much higher than all these people outside of the clusters. Be careful. We got this cluster only using the metabolism data sets. We didn't even use the medical profiles. we cross-check every person's medical profile, compute the distance, also compute the risks, and notice that all these people getting the disease, the risk is much, much higher than all these people. There are actually 4,000 people inside here. There's actually 46,000 people outside here. The average of all these people getting whatever diseases, the first 50 type of diseases, is much higher than the surroundings. So we thought learning the stuff from this metabolism might be useful in terms of profiling the whole populations. Of course, I think for the biostats people, maybe you can do better, but this is something I wanted to share with you guys. And since I'm already run out of time, then I will somehow stop right there. And thanks for your attention.
I think this is a very fascinating result. You got this, so the one-dimensional trajectory. Follow-up question is there any meaning about the ordering? Maybe the disease was, developed earlier, that sort of trajectory.
It's being investigated. We just got this results half a year ago, but we're still looking at these things. Yeah. I don't have too much domain knowledge about all these Euclidean things. Yeah. Someone in my group knows a great deal of these. Yeah. But I thought this was very inspiring because we only used the metabolism to do the classroom and then cross-checked with the medical records and found the difference.
Did you, in the end, do you know anything about the biomarkers that you found? Like, or is that information still being processed?
Do I know anything about the biomarkers?
Yeah. So you said that there's clearly something in the biomarkers that's being correlated with higher rates of disease and so on.
So
Do you have information that you can now tell the medical people these kinds of properties or?
So that's right. So what I did was actually there are actually totally 251 biomarkers.
Yeah.
So we somehow grouped them into 7.
Yeah.
I mean, we could have just fit the manifold fitting onto this whole 251, but we somehow grouped them into 7 different pieces.
Yeah.
Then we fit the manifold independently, separately for each of them. Maybe I think that within the C1, C2, C3, or whatever C7, some of the, for example, in the C1 here, okay, these variables might be, for example, I actually got this 2 cluster things. These are the blood sample from the MRI stuff.
Yeah.
So they might tell you a lot about some of the diseases, but I don't know about.
Oh, that's what I was asking.
Yeah.
Whether you have sort of information about which are the active.
I don't know. For this is
Interoperability.
Yeah. So I mean, the interpretation presumably at some point is being attempted. I was just wondering
Yes.
if you knew anything about.
I don't. I don't. But we're still pursuing some of this.
Yeah.
Because I didn't sign up this UK Biobank just for publish a few papers. We actually wanted to know more about it.
Yeah, I also think it's astounding that when you do all this and you reduce to dimension 2, you have something that's got only 1 dimension, which means that intrinsically up there, it's really only dimension 1. If you see a projection to something 2-dimensional, it's really 2-dimensional, but you don't know what dimension it was up there. But if you have something that's projects down to 1 dimension in 2 dimension, then it was 1-dimensional all the way up there. That's kind of amazing, right? It's like, why else would you'd be projecting away? But you'd be projecting away the variation. Which is the point of not doing that. Presumably the point is to not do that.
This just sounds speculation, and maybe it poses some sort of
Okay.
carriage for you. So suppose that I have a pure random data.
Yeah, pure random data.
And then you can usually, you can use this space, what do you call it? Space filling curve. Try to do it this way and fill everything. So the fit seems to perfect well, but it's not leading to anything, right? I mean, space filling curve.
Yeah, space filling curve.
Yeah. So how did you avoid that?
I don't understand the question. How did I
Well, you got
avoid?
a dot dots there.
Mm.
So looks like it's a two-dimensional uniform distribution. But if you are really careful, you can trace the nearby and go this way, this way, this way, this way, and that becomes a one-dimensional embedding.
Yeah, that's embedding.
Yeah,
Yeah.
but that does not make sense.
We didn't get embeddings.
Okay, so
Yeah.
you did not.
We didn't get embeddings. We didn't embed things. This is just for visualization. We learned the manifold actually.
Yeah, but the thing is that when it's two-dimensional manifold or just the one-dimensional manifold.
You never know. You never know.
Okay, so
never
you
know.
just do your best.
Yeah.
So actually. it
I
could
actually
be-
don't need to know the dimension of the
Okay,
manifold.
so actually, it could be just a two-dimensional manifold, but you just sort of try very hard, so it becomes a one-dimensional. So it looks like you can reduce two-dimensional to one-dimensional, but it's not really reflect the truth, right?
Well,
What are the manifold dimensions you ended up with?
I don't need to know the manifold dimensions.
No, I'm
Can
not
you figure them
criticizing,
out?
I think the
Mm.
result
Can you
is
figure them out,
very
yeah?
good, yeah, but.
So you presumably did fit something.
Yeah, I did something.
So is it, can you then in the end read off what that dimension was? Like, I know you don't need to know it beforehand, but afterwards you probably fit something.
You can actually,
What's it?
one can actually check the local map,
Oh.
local graph
Yeah,
to
yeah,
to
yeah.
compute the
That's what I'm
eigenvector.
asking. What was that dimension? And if the dimension wasn't one.
We didn't
what
check for
was-
the metabolism, for the single cells, okay, even if it's 15,000, the local map, the local, I mean, the covariance matrix, if you do the single variable decompositions, it won't exceeding, let's say 15. It's always small.
Guess my question is about the next slide. We go to the next
The
slide.
next slide,
So if the summary on the right, in box C,
yeah,
if the manifold that you fit wasn't dimension one, then what justifies this being a reasonable visualization of something that isn't dimension? That is, I would really need to know what dimension was the
Well-
manifold was end up being fit there, because if it wasn't one, then this is misleading to me.
But so
Because to me, like, you can't summarize a two-dimensional manifold in a
So you
one-dimensional.
cannot
manifold.
really answer the question unless you have a sort of a structure embedded, so that you can have a meaningful way to see whether it's one dimension, two dimension.
someone has to
So
check.
This
someone-
one is just, the way I said, it's just space filling, but if you have embed something outside of that, then you can use that matrix to
But-
quantify
Yeah.
what it
But
is.
the point is that something was fit. So I just want to know what dimension that was.
Wait,
Or you can-
someone has to check, I mean, one
Yeah,
can check.
you should check and
Maybe I check.
then label it, but that's, we know.
Okay.
You also had a slide about Colloquium fitting, which you skipped. Can
I-
you say something about that as well?
I actually hate to comment this, but that was for simulation, okay? Because you write a paper with Yao and he wanted to do something. We checked it. So the Colloquium Yao. Well, this is simulation because we actually know the manifold. So we add some data onto, so we actually add some noise onto this. In the ambient space where the manifold was actually embedded, we actually add some noise on it. So the fitting was basically we'll do is push all these points to this underlying manifold where we already know because we know the equations. And then these are all somehow, all these 1, 2, 3, 4, whatever 8 methods claiming you can do fitting and then try to compute the distance between our fitted points with the underlying sort of things. Yeah. And these are the things show you, for example, my method, which is, this is the input method. This is my method. These are the all, some other method which has been published in the PNS. And then compare for example, this shows you, for example, the box plot is actually, lower than these, meaning that my method is better, that's all.
This is a two-dimensional manifold and then
It's 4-dimensional.
four-dimensional.
Yeah, this is just a slice of the visualization in 3-dimensional space.
When you partition into 7, they use 200 people one partition into 7 parts. So how you get this partition on the other?
Good question. Think about the matrix is n by p. P is 251, right? So what my postdoc did was trying to measure the distances and try to split them. But if you transpose that matrix, apply the fitting on the transport matrix, you actually can separate them. Do you see what I'm saying? Your question was, how did I split the 251 to 7 different classes, right? I can say you can do the transpose of my data matrix, apply the fitting once, and you get 7 different classes. The X matrix is 200,000 multiplied 251. If you fit the manifold, if you do the fitting on the X transpose, you're actually splitting the variables, not splitting the samples. So these variables will become the different groups. Yeah. We can also fit the manifold along in it to get to just solely on the 251, but my guess is the performance won't be too good. Yeah, because this leads to another question. People say, what are is it possible to extend the fitting to multiple manifolds, or the manifold where the points are not uniformly distributed? But that's another ongoing things, which I don't have any answer to. Yeah.
There are no more questions. Let's thank the speaker for a great talk.
Great.